Neutrophils play an important role in chronic inflammatory conditions and acute viral and bacterial infections in the lung. In order to study the mechanisms of neutrophil trans-epithelial migration into the airways of children with severe RSV bronchiolitis, we've developed a method using epithelial cells grown from nasal brush biopsy. Our gravity-fed system supports neutrophil migration in a physiologically relevant basolateral to apical direction. This technique allows us not only to measure the numbers of neutrophils that migrate through, but also to observe neutrophils migrating through the airway epithelium under the microscope. We can also collect the migrated neutrophils and the airway epithelial cells for downstream analysis.
Here we found that RSV infection led to an increased recruitment of neutrophils across the epithelial barrier. We also found that neutrophil adherence to infected epithelial cells was significantly increased following trans-epithelial migration in RSV infection. These migrated neutrophils showed a unique phenotype in regards to RSV infection. We found increased expression of CD11b and neutrophil elastase, which were higher on migrated neutrophils compared to neutrophils that migrated in response to mock-infected epithelium. Interestingly, we also found increased expression of these markers on neutrophils recovered from the basolateral side compared to our control condition of neutrophils exposed to media alone. In order to investigate whether this was due to secreted factors from the basolateral side that were changing the phenotype of these neutrophils, we also grew airway epithelial cells on inserts with a 0.4 micron pore size. This pore size prevents neutrophil migration, but would allow neutrophils on the basolateral side to be exposed to secreted factors from the airway epithelial cells. We found that under these conditions, the same increase did not occur on the basolateral neutrophils when neutrophils were prevented from migrating, suggesting that activated neutrophils reverse migrate from the airway to the bloodstream side, as has been suggested by clinical observations. We then combined our findings with a temporal and spatial profiling and suggest 3 initial phases of neutrophil recruitment and behaviour in the airways during RSV infection. 1. Initial chemotaxis. 2. Neutrophil activation and reverse migration. 3. Amplified chemotaxis and clustering, all of which occur within 20 minutes. This work and the novel outputs could be used to develop therapeutics and provide new insight into how neutrophil activation and a dysregulated neutrophil response to RSV mediates severe disease.